Hello everybody, get ready to strap yourselves in, including you who's about to jump out of a parachute. Oops, too late. It's the 6th episode of Biology from the Bedroom with me, Benito Wainwright. And what a time we've had so far. Up till now, we have learned that freshwater ecologists laugh like this. Sensory ecologists can sing the endings to Girls Aloud songs. Oh. And evolutionary biologists are social parasites.
Gonna take a lap to drag me away from you.
Today's guest is Mr. Jack Greenhalgh, an aquatic biologist from the University of Bristol. After graduating with a degree in marine biology from the University of Bangor, Jack went on to complete a Masters in Aquatic Science from University College London. Now for his PhD, he is investigating the use of non-invasive methods to detect invasive species. In particular, the signal crayfish, an extremely destructive alien species in the UK. So, are these non-invasive techniques any good? Or is this just Jack's way of saying he's not doing anything at all? Well, without further ado, it's time to beam potential science leader of the future, Jack Greenhalgh, into my bedroom. Hello Jack, how are we doing?
Hi, how's it going? I'm great, thanks.
I'm very well, thank you. How are you coping with the lockdown so far? Is it treating you well?
Yeah, fairly well. Very lucky to be living in Bristol, really, because there are so many really lovely little nature reserves that I've been exploring on my bike. Many of which I've got great ponds in. So.
Oh, I love a good pond. But if you were to... If you were to summarise this global pandemic that we're experiencing at the moment in maybe just one word, Jack, what would that word be, maybe?
Unprecedented.
Unprecedented. That's a good one. That's a good one, to be fair. I'll add that to the list. Jack, before we start talking about your PhD stuff, you, as I said earlier, did a degree in aquatic science. So... that seems like quite a niche area of science. So why did that course particularly whet your appetite, if you pardon the pun?
Well, it's a niche area of science in a totally strange area to be doing it in the centre of London, studying ponds and rivers and things. My final year project was my introduction really to bioacoustics, which is this study of sounds that animals produce. And the title was recording the underwater sounds of ponds, and when I saw that the first time, I thought ridiculous.
Because at the time, had you not, heard what a pond sounded like before?
Absolutely not, no idea. And it always amazes me every time I go to a pond, because you look at it and you think this isn't going to work, is it? Isn't going to be anything in there? And the moment that you put the hydrophone in, this underwater microphone. Well, it just comes to life, Benito. It's like an underwater disco.
An underwater disco.
All kinds of things, buzzing and whirring and popping and gargling.
Goodness me! What? Lady Gargle? Is that what was playing?
Lady Gargle, absolutely. Yeah.
Indeed. How interesting. So that's kind of led you into your PhD research, which initially started out as looking at invasive species, in particular the signal crayfish. Do you want to tell us a little bit, just as an introduction, the story of the signal crayfish and why it's such a massive problem in the UK?
Yeah, sure. So the signal crayfish is native to North America, and it was introduced to Europe as food, really, for monks and things like that. But they had their little ponds. And they would stock them with these different types of crayfish, primarily the signal crayfish because it was larger and it grew faster. Over time, these species gradually took over Europe because they have these qualities of being highly fecund and growing quickly and being tolerant of different environmental conditions.
Right.
And not only that, but the signal crayfish also carries with it the crayfish plague. Really, actually, rather than the signal crayfish itself, it's the crayfish plague that it carries that's caused such a dramatic decline in native crayfish species, notably our white-clawed crayfish, which is our native crayfish species, which has declined by about 90%.
That's the problem with the signal crayfish, I suppose. It's just very shellfish. But so how are you applying the techniques that you learned previously on bioacoustics to study how the signal crayfish spreads, if you like? How do those two topics kind of link together?
First of all, we know that crayfish produce sound, and like other aquatic arthropods, they produce sound by this process called stridulation, which is a great word.
Yes.
But basically what it means is producing sound by rubbing two hard body parts together, kind of like crickets do.
Right, we've all done it, yeah.
We've all done it. So the idea is, can we record different species of crayfish, say the invasive signal crayfish and the native white-clawed crayfish, separately in recording tanks?
Right.
Listen to the sounds that they're producing, and then characterize those and use those sounds to detect each of these species in the environment.
So you've actually recorded some cool stuff here. Now, it's quite an interesting sound, and as I say, it's probably not something which our listeners have heard before, so it would help if you talked us through what we're actually listening to here, if that's all right, Jack. So this is Jack Greenhalgh's latest release from Old Sneed Park.
Well, here we go. You can hear all kinds of different things in here. So there's the noise of aquatic insects, and they're making those sounds by stridulation.
Right.
They kind of sound like, do you ever have one of those wooden frogs with a little wooden thing that you could scrape over the top? Aquatic insects tend to sound like that. And then occasionally you can hear gas bubbles.
Oh, don't tell me what that is, Jack.
I don't think you were swimming in the pond at that particular
Oh, right. Okay, right.
Those gas bubbles are caused by decompositional processes.
Nice.
As they kind of come up from the sediment and pop on the top.
Wow, that's amazing. I mean, that's what you pay your taxes for, isn't it? The sound of a pond. Unbelievable. So, how is what's going on in the world at the moment? This COVID-19 pandemic, which you might have heard of. How is this affecting your research plans at the moment, Jack?
So I've been cycling to all these different ponds that are around in Bristol, and there are loads, and just throwing the hydrophone in, leaving it for a week, and the idea there is to try and characterize the soundscape, so that's all the acoustic signals present in the environment, and see how long do you have to record for in order to capture all of the acoustic diversity that's present within that pond. And also what I'm trying to do is see what kind of day-night cycles are there.
Okay, I mean, I assume in the nightlife in the ponds, is that where it's at?
Yeah, well, I mean, it's not a disco for no reason.
Yeah.
The sun goes down and all the stridulation starts.
Right, moving swiftly on from that. Also on Biology from the Bedroom, we want to talk about times where things haven't quite gone to plan, if that's all right. So times where our guests have been made to look like an idiot, quite frankly, or when things have just not gone very well at all. And I think this is something which a lot of postgraduate biologists can relate to. So tell us about this story of when you were stuck in a van, Jack, 'cause that seems like quite a perilous event.
So our PhD cohort, we were on a training thing in Wales in the Brecon Beacons. The director of our PhD program said to me, "Just go and collect this piece of equipment from the van." Okay, fine. I'll go to the van, open the door, climb into the back of the van, and the van was parked on a bit of a hill. The van was facing down the hill, so I get into the van, and the door slams behind me shut, and I don't have my phone on me for some reason. It's pitch black in there, and I spend the next 10 minutes fumbling around in the dark trying to find the door handle. And then at that point, the director of the program, opens the door of the van and finds a very sad,
skeleton,
lonely Jack Greenhalgh in there.
Oh, goodness me. Was it quite embarrassing?
It was pretty embarrassing, yeah.
Sure.
She said, come on, get out.
It's time to put our guest through their paces. So over the past 5 weeks, we've been on a mission to find Britain's most promising young research biologist. And in order to find that out, we have to put you through, Jack, the cryptic question clock quiz.
Oh, cryptic question, where's it gonna be, where's it gonna be? Cryptic question, where's it gonna be, where's it gonna be? Cryptic question, where's it gonna be, where's it gonna be?
This is a section of the show where we get our guests to answer as many biology-related questions in 60 seconds as they possibly can. But there's an added twist, and that is the cryptic question.
Oh, cryptic question.
What's it gonna be? What's it gonna be?
One of the answers to the question I'm about to ask you is also an answer to a question in a non-biology related area. Now, to be honest with you, as I said last week, we've received a lot of complaints about this, Jack, to be honest with you. People saying
Well, I'm
that
not surprised.
the cryptic question is too difficult. Last week we tried to make it easier, still complaints. So I promise you, this week, Jack, we've made the cryptic question as transparent as possible. Okay? As transparent as possible. If you're able to spot the cryptic question, you have to let me know using your signature buzzer. And this week, your buzzer, Jack, shall be the sound of Craig Greville Horwood being nipped by a signal crayfish. Can you give us an indication of what that might sound like, please?
Ah, you bugger! How about that?
That's good. Yeah, perfect. And today, that cryptic question topic shall be famous figures born in November in the 1600s. Okay? So, Jack, are you ready to face the cryptic question clock quiz?
Oh, yes, go on.
In that case, your time starts now. What liquid do fish typically live in?
Water.
Correct. Generally speaking, what are microscope slides made out of?
Glass.
Correct. Which intermolecular bond is formed between complementary base pairs in DNA?
Covalent.
Incorrect. Name 3 species of British tit.
Blue. Great. And, I don't know, pass.
Small circular pieces of DNA found in some bacteria are known by what name?
I pass.
The 3 domains of life consist of bacteria, eukarya, and what other prokaryotic group?
Archaea.
Correct. Name a type of cell that you would find in a leaf.
Plant cell.
Incorrect. Individual optical units in an arthropod eye are known by what name?
Oh, dear. I don't know, pass.
What type of animal is a hellbender?
A lizard, is it?
Incorrect. So at the end of that, Jack, you've got 3 points, I'm afraid, which doesn't put you at the top of the cryptic question clock quiz leaderboard. Unfortunately, you weren't able to spot the cryptic question. Now, the second question I asked you, Jack, was, generally speaking, what are microscope slides made out of? And you said...
Glass.
Yeah, so glass is also the material which Prince Rupert's drops are made out of. These are tadpole-shaped beads named after Prince Rupert of the Rhine, who brought them to England in 1660. But I did say, didn't I, that I would make the cryptic question as transparent as possible. Oh, never mind. That's why it was glass. Never mind. Well, whilst you're recovering from that, Jack, we've got a special treat from you. Here at Biology from the Bedroom, we've managed to track down the very people who inadvertently released the signal crayfish into the UK. And we found out that they've actually recorded a special apologetic single for you. Here it is. Oh baby, baby, how was I supposed to know that something wasn't right here? Oh crayfish baby, we shouldn't have let them go. It's not the way we planned it, honestly, we were simply crazy. Tell me baby, 'cause I need to know now. Oh, because their invasiveness is killing me, and I must confess I still believe, when you're not with them, you'll surely thrive. Put down your sign. Let's put a stop to crayfish crime.
Disgusting!
Anyway, we're getting up to the end of the show, Jack. If you were to... So we're going to end the show on a nice song, I suppose. I think that's something which we always like to do. Of course, there's a limit to the songs which you have available to us, unfortunately. But what song would you end the show on, and why, Jack, in your opinion?
Well, I'd like to stay in touch with the biological theme and go with Chameleon by Herbie Hancock.
Nice. Let me just check. Unfortunately, we don't have copyright permission to that song this week, I'm afraid, Jack. The only song which we had copyright permission to this week was Cry For You by September. Never mind.
Right, okay.
But we do have a very special treat for you, Jack. So,
Yeah.
Did you know that worldwide pop sensation Rihanna is actually a massive fan of the field of bioacoustics? Did you know that?
I didn't know that,
No,
no.
you didn't know that. In fact,
That's great.
it was a close encounter with a mayfly larva in Barbados, which inspired her to write her debut single, Pond a Replay. Right, so since we found out this information, we obviously got in touch with Rihanna's team and have organised a very special live stream for you. So here is that very special live stream.
Oh, that was great.
That was lovely. Well, thank you so much, Jack, for coming on Biology from the Bedroom, and best of luck with the rest of your research.
Thank you very much. You too.
Thank you very much for listening to this podcast. If you liked it, don't forget to subscribe to the channel for even more wildlife and science content. I'm going to take a bit of a break, but never fear, I'll be back soon to put even more perky PhD students through their paces and find out who is the most promising research biologist in the country. In the meantime, I need to tell my pet signal crayfish to stop holding up some traffic. See you soon, everybody!